All right. Hi, everyone. Welcome to our first interview series of StatsUpAI Corner with Wenyi. So I am Wenyi Wong. I'm the program chair of StatsUpAI, which is an official interest group of the American Statistical Association. And I'm also the host of at least the first few episodes of Corner with Wenyi interview series of the Stats of AI. I'm also a professor of in the Department of Bioinformatics and Computational Biology at the University of Texas MD Anderson Cancer Center. Okay, all right. Oh, can't move this slide. All right. Okay. Can you guys see my slides okay?
Yes.
Yep. Yes.
First is StatsUpAI's mission is to organize activities that empower statisticians to participate more actively in AI research and leadership. And this initiative is dedicated to enhancing the role of statisticians in addressing real-world challenges through AI research. We emphasize the dual importance of leveraging both statistical methods and AI tools, ensuring that statisticians are not only participants, but also influential leaders in applying these combined approaches to solve practical problems. We're proud to announce the launch of our brand new Corner with interview series. With the goal of making conversations with leaders in statistics, exploring how rigorous statistical thinking is shaping the future of AI, one dialogue at a time. It is my great honor to introduce the featured guest of our very first episode, Professor Wong. Professor Wong is currently the Pierce Family Goldman Sachs Professor of Statistics and Biomedical Data Science at Stanford University. He was elected to the member of the National Academy of Sciences of the USA in 2009 and the Hong Kong Academy of Sciences in 2015. He also won the COPS President's Award, the highest honor in statistics, in 1993, and the COPS Distinguished Achievement Award in 2021. And Wing has made stellar contributions to statistical theory and also to modern genetics-based biology. Like R.A. Fisher, the founding father of modern statistics, Wing Wang is a theoretician, but with a knowledge of science and particularly biological science, which has an unusually broad impact. Wing is one of the most influential figures in modern statistics and computational biology, and Wing's pioneering work on partial likelihood, semi-parametric maximum likelihood, data augmentation, and Monte Carlo methods has profound impacts on likelihood influence, Bayesian statistics, modern machine learning, and artificial intelligence. His style of research, with astonishing depth, breadth, and originality, has influenced many statisticians, including not only his students and postdoctoral advisees, but also his many colleagues and generations of young researchers, including myself. I'm also very happy to, and it's a great pleasure to introduce our two discussants of the day, Professor Jing Liu, the Tsinghua Distinguished University Professor, Chair of the Department of Statistics and Data Science at Tsinghua University, Professor Emeritus of Harvard University, and a member of the National Academy of Sciences of the USA. He studied his PhD with Wing Wang and Augustine Kahn in 1988 to 1991 and worked on theory about sequential importance sampling and Gibbs sampling. I'm also very happy to introduce Professor King-Fai Oh. He's a professor of computational medicine and bioinformatics at University of Michigan, Ann Arbor, and he received a bachelor's from Tsinghua University and a PhD in structural biology at University of Oxford, and he completed his postdoctoral training with Wing at Stanford, where he developed the earliest bioinformatics methods for long-read RNA sequencing data. And hi, everyone.
Hi.
And yeah, so it's great to have you all. And so Wing, have you met, have you talked to your former trainees recently?
Well, I saw June just 2 months ago. Um, Keen Fight, I don't know, maybe more than a year. We have not talked
Yeah,
for more than a year.
I met you during the symposium at Stanford about yeah
Yeah,
almost two
that was
years. Yeah, two
more
years.
than 2 years. Yeah.
Great. Yeah. So it's my great pleasure to have you all here again, and consider this a reunion in a relaxed environment, and hope that we can have a great conversation. So I have a list of questions that'll cover four parts. The first three parts will focus on your career journey when so focusing on the past in the part one, the current part two, and then the future part three, and then the part four will be question about and then for you to share your advice on mentoring and the future generation. Are we ready?
Yes.
Yeah, great. Okay, so looking at your career journey, which started from Chicago in 1980s, and then move on to Chinese University of Hong Kong in 1990s, and then back to the USA to UCLA and then Harvard in the 2000s, and then moved to Stanford. I started your professor position there in 2004, and then has been there since. So let's go back to the 1980s. So in the late 1980s, when you and Martin Tainer introduced your foundational work on data augmentation and MCMC, did you sense that you were opening a completely new chapter for the field?
Well, not really. I mean, in those days, I think the Bayesian statistic area, people have been focusing on using numerical quadrature and asymptotic approximation to extract information from the posterior distribution. So I was very interested in this topic because I was reading paper by Dennis Ningley and Adrian Smith. They're all saying that this is very important. And then at that time, I was also reading the paper by Dempster, Lott, and Rubin on the EM algorithm. And Martin Tanner, who just got his degree from Chicago and was working in medicine. And he and I often discuss these topics, and we thought, well, maybe we can put these two together and try to come up with something new for Bayesian computation. So we managed to sort of exploit the structure inherent in this complete data formulation of the EM algorithm and use it to create some algorithm for doing Bayesian computation. So 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 that immediately make the method sort of useful for many sort of familiar parametric problem that people have been looking at using the EM algorithm. So that familiarity, I think, help people to to understand and appreciate the thing. To personally for myself, I think the the feature that I think is somewhat new in that paper. I mean maybe people have been using that, but it sort of appeared about the right time and catch people's attention. Is that we are basically saying that you instead of trying to do integration to get marginal distribution or expectations, you aim to generate a sample from the posterior distribution. And then with that, you can, do your exploratory and data analysis or do marginal histogram. And basically, you can get any information you want if you have a good enough sample. from the posterior distribution. So that sampling perspective, I think, is somewhat useful and maybe partly responsible, but for why the paper will have some impact. Yeah, so
Yeah.
we have no idea that it would it would become a very popular method, but we think it's a fun thing to do,
But
basically.
it is a discussion paper, though. So it's very rare to have a discussion paper in JASA, even
Yeah,
now.
I was surprised. Yeah, I was surprised.
Yeah, I guess someone must have seen that. There are quite a few stories since I was just after that paper, I just entered the University of Chicago. I think
Mhm.
it's a prime time to actually get to be exposed to that problem.
Yeah, and then and then follow up. So what do you think? What it was the paper that's the most cited from this line of work?
I think Alpha Smith is very highly
Yeah,
cited.
Goldman Smith definitely. Yeah.
Yeah. Wynn's paper is very well respected. I think it's not the most highly cited after Alpha
No,
Smith.
no, no.
All right. Yeah. Great. Thank you. Next question. Moving on. Yeah. So you received the COPS President's Award in 1993, and then the Distinguished Achievement Award in 2021. Um, essentially bookending a career of massive influence. So over those decades, as you worked on a wide range of topics from foundational theory all the way to computational biology. So how has your definition of impactful work evolved? And what drives you to keep crossing into new territories?
Yeah, so I think whether the paper will be impactful, as I said, is partly luck, right? But I believe that you want to work on a problem that people care about. Have either it has some deep scientific question behind it, or maybe it's a kind of a difficult open problem in mathematical statistics. So if you manage to solve those problems, then definitely you have a chance to be impactful, but that still depends on luck, I think. Um, and so I mean, I wanted to work in biology with the idea that we want to contribute to the development of biological science. But I have to say that you know that goal has not been achieved. But you know we managed to contribute some sort of useful statistical method to analyze some biological data, right? Like microarray. So the paper with Cheng-Li, it's very proud of that. But
Yes.
it's not really solving a biological problem. So, in recent year, my desire is to make an impact in solving a biological problem, but that has not has not become a reality yet. So still trying. Yeah.
Yeah, so luck aside, right? Your drive, it's just what drives you? It's
Well,
it's
it's
amazing.
interest. I think interest is everything. Yeah, if you have interest, you will work on something, day and night and try to make progress. Yeah.
That is truly amazing. Yeah. Um, so and then related to that, right? So when you were already a full professor in statistics at the University of Chicago, and then you decided to devote your time in the early 1990s, right, to learn about biology, and started. I heard the story on the street is you took a undergrad textbook on biology and read the whole book page by page. First, I would like to confirm if that's true.
That's more or less true. Yeah. It was Jim Watson's molecular biology of the gene. So it's a multi-volume book.
Yes, that's
It's very
classic.
well written. It's very, very easy to get engaged and
Yeah,
very
I do remember those books in Wing's office when I came to Wing's office for my interview, where I interviewed for
Mhm.
my position. What happened? I find okay, there's quite a few biology books there in the bookshelf. It really surprised me.
Yeah.
So, yeah.
Yeah, I think that's kind of very inspiring because that's what the most lacking in most like mainland Chinese education. Sort of we were more geared towards solving homework problems or like 刷题
Mhm.
or something like that. Now it's even worse, right? In my days,
Mhm.
it's much better, but even so, it's kind of very hard to imagine it, to start some subject completely anew with no background. I think that's very inspiring. That actually motivated me to look into biology to actually search for some challenges in computational biology. Otherwise, I would never do that. I would just do some familiar like mathematical stuffs and so on.
And was there any specific event that triggered the beginning of reading an undergraduate textbook? Just curious.
yeah. I mean, there was this paper, written by Kerry Mullis, who was the inventor of the polymerase chain reaction.
Hmm.
So he has a paper in Scientific American.
Mm.
I think the title was like The Unusual Origin of the PCR.
Hmm.
And so I remember reading it, and the concept of PCR is, involves some mathematics, right?
Hmm.
Exponential multiplication. And so I think, that was really elegant. I did not have any biology background. In my college days, I did not take any biology courses, even not even biochemistry or organic chemistry. So,
Yeah, me either.
but that paper made me realize that maybe, biology is not such a boring field, right? So then I start to read up on genetics and cell biology. I find it's really a very rich and rapidly developing, very interesting area. Yeah. So that paper really changed my trajectory. Yeah.
Wow, that's so amazing to hear. And when you decided to change, have you does it cross your mind about the career stage or how yeah, like other like promotions?
Well, I was already tenure at that time, so I wasn't
Okay,
worried about promotion. But
perfect timing.
I think from the time I start to read up on biology to the time I publish my first paper is probably more than 6 or 7 years,
Mhm.
right? So,
Yes,
so
yes.
when you start, you don't really plan that far ahead. Yeah, it's mostly driven by interest.
Yeah, wonderful. And follow up on that, you were only also one of the first mathematically trained statistical scientists to take up running a wet lab as doing as well as doing the computational and theoretical studies, which happened in around 2005 after you have moved to Stanford.
Hmm.
So this is again truly inspirational for many of us, and now more and more, actually, computational scientists have wet lab. So what is it, from your perspective? What is that the wet lab can provide that computational work cannot?
Well, as all of you who collaborate with biologists would know, they are very they're very focused, right? Usually each lab has a focused problem that they're trying to solve. So you
Mm.
look at their data and you say, well, there's a gap in this data. You may want to, do some experiment and generate this particularly informative type of data. They would not listen to you basically, right? So you feel that you sort of know, there's this type of data that will be useful, and you want to be able to generate it. So that was the motivation, and because of the advances in instrumentation and so on, so a lot of the data are not that difficult to generate. And so I thought, well, may give it a try. But the fact is, I have the wet lab, and we have generated some more or less useful data, on embryonic stem cell development and so on. But the result was not what I expected. We didn't make any discovery in true discovery in biology. I think it's just, but it's helpful for our lab member to sort of understand the whole process. And so I think there's some indirect benefit, but I would not say I have achieved my goal of setting up the lab. Yeah.
I see. Yeah, so and then can you there's an important so we talked about the study in the lab, and another important transition or beginning is the bioinformatics microarray analysis of microarray data.
Yeah.
So can you take us back to the beginning of that? How did that start?
Well, so I was in Hong Kong, I think, in the period from 1994 to 97, and that was after I have spent some year reading up on genetics, and so that got me interested in the method of doing sequencing, and so I was trying to come up with some method to do sequencing using some concept like the microarray. Basically, those are we call spotted arrays on cellular membranes. People just print DNA. So you take a plasmid and you print it on the array. So you have these DNA spots, and then you hybridize to it, and then you do imaging. And I was thinking that, well, maybe we can use this for doing sequencing by taking size fractionated fragments of DNA and then put it on the spotted array and see which array light up, and then trying to reconstruct the sequence. And then we can do it in parallel. So one electrophoresis would generate several hundred sequences. So I was thinking about the something like the microarray. So then one day I pick up the issue of Science, and I saw two papers. One is Pat Brown's microarray paper. The other one, I think, is the SAGE paper. There was the Johns Hopkins group and Vocal Science group. They were doing SAGE. So it's a two parallel paper that propose methods for doing RNA analysis, basically. And so I look at that, I say, wow, this is really exciting. So I immediately try to think about the statistic associated with that. And then 1 year later, Affymetrix commercialized their way, and then this kind of data start to appear. And so we were early in working on this problem, sort of because I was kind of thinking about microarray in some sense, not for doing gene expression. I was thinking about it as a method to do parallel sequencing. Actually got a US patent on that method I proposed. And later on that was licensed for a while to Affymetrix, I think. But it was the gene expression application that really made microarray very popular. And so we're lucky that we are aware of the usefulness of this kind of approach, and then also fortunate enough to see some of the early data generated using Affymetrix array. So So that was that was my story, how I got involved in that. Yeah. So I think that all those time spent reading up on biology was not wasted because it sort of prepare you so that when the opportunity presents itself, you would be the first to recognize that. Yeah.
That's amazing. And I can tell you that since I did my postdoc at Stanford Genome Technology Center with Ron Davis, and from my side of the story was when they generated the first set of microarray data, they don't know how to analyze it, so they send one copy to you and one copy to Terry.
Yes. Yeah. Yeah. Arithmetic came out of Ron Davis' lab, I think. Yeah. That's right. Yeah.
So about this sequencing technology idea, and you were in it from the very beginning. Can you walk us through the whole journey of what are the kind of the evolution of the technology and what you think of the current and the future in terms of sequencing technology and statistics?
So, in the early days, it really is a very slow process where sequencing you put the DNA fragments through a electrophoresis. One channel give you one sequence. So in the very beginning, I sort of thought that one has to go parallel. And so that idea I just discussed that as a pattern the pattern was called, parallel oligonucleotide sequencing. But it turned out that that method was still depending on electrophoresis, although the fragments are put on microarray. So that turned out to be not the most effective way to do parallelization. So later on, I think George Church lab, and then there's a company in the Bay Area. I forgot the name of it was acquired by Illumina eventually, and that become Illumina's sequencing technology. So their idea is to use imaging basically.
Alexa?
This short read sequencing is massively parallel, and that turned out to be sort of the right way to do the parallelization. So I think that technology really is a breakthrough. I mean, it just changed everything. And we don't know what will come next, but I think even in its present form, the technology is already so immensely powerful that I think we are still far from exploiting the full potential of what it has to offer.
Yeah, absolutely. Kim-Fai and Jun, do you have anything to add to the part one, or shall we move on? Yeah.
Jun, you may go first.
Go ahead. I have quite a bit.
When you mentioned the technology-driven research in the Wong lab, yeah, I can tell as well. Definitely, the Wong lab was always in the front lines of using or analyzing the newest technology. When I joined the Wong labs in 2008, 2009. By that time, Illumina sequencing and the other short read sequencing come out, right? And I think Wing and the whole lab developed almost the very first series of the software to analyze those data since the Hui Jiang, and we developed I would say that with the very first statistical model for the RNA-seq quantitative analysis, that is a Poisson model, right? I recall after many years ago, many years later, when I read some review article, they still cite that is the very first statistical model for the high throughput RNA sequencing. Yeah, of course, we know the D chip, right, for the microarray. Yeah, of course, and well. When I established my independent projects in the Wong lab, we started to analyze the long read sequencing. I think, I think the new technology always provides some new information. I think Wing always encourage us to explore the potential in this new technology. This is something that my own lab still working on now. When we foresee the sequencing technology, I heard that Oxford Nanotechnology are developing some technique for protein sequencing, right? If we can do protein sequencing, well, we will have a lot of opportunity to study protein in a very high throughput way. Right? So yeah, let me just and here, so Jun.
Yeah, well, I think I'm always very impressed by Wing's willingness and the capability of getting into anything really new. So it's pretty amazing to me. I think the first time, like my entry into computational biology, I was purposely trying to be a little different from Win and Augie because Augie has been working on genetics, and Win at that time was interested in protein folding problem. Although I eventually became quite interested in that problem, but at that time I thought Win has been working on that. If he couldn't work out, then I probably should not get it. So I actually started with the sequence analysis. I first met Chip Lawrence and tried to do sequence analysis. It's but Wayne was the first to actually advise me or chatted me or allude me that maybe this chip technology is interesting. I should look into that. I remember I took Wayne's advice. Actually, I knocked at the door of Pat Brown's lab, and he wasn't there. So then that's the end for me at
Yeah.
that time. I sort of feel sorry I didn't knock another time, but I came back like 5 or 6 years later, following Wayne's work later on. But it's already one step behind. But it's quite interesting. I've been also looking at and following. Uh, I think it's hard to follow all of Wayne's lead, but one or two would be good.
Yes,
So it's
we need a large quantity of people
so it,
follow with. All right, great.
Oh,
So moving on to part two, current. We're at the current, and then let's talk about AI revolution. So wait, so your lab recently developed deep learning tools like Epi-PRS and Epi-GPT. So when faced with a complex biological data set today, how do you decide when the problem calls for a deep learning architecture versus a more traditional, interpretable statistical model?
well, I think if you have a lot of data and you believe that there is regularity in that data that you can exploit, then this kind of deep learning
No brainer.
type AI model
Mm-hmm.
could be very powerful. And of course, you also have to have the computational resource to do
Mm-hmm.
that. So our experience has been that these models are really quite useful, given you have the correct data. But there are many situations when the information that this model can provide is only uh, very general. It is not specific enough to answer the question that you really care about, right? So yeah, recently we've been working with a lab that is interested in designing, a protein that has certain intracellular function. But what they have is a version of the protein that can function extracellularly outside the cell. So so they want to redesign the sequence so that it can function intracellularly. So it turned out that this protein language model cannot generate that because they are trained basically to generate sequence that would lead to stable structure that would fold and that may be soluble, and they don't they do not have any way to give the protein the specific property that you want, such as that is intracellular. So then once you focus on a more specific problem, then you find that you have to combine your large language model or the foundation model with some more specialized model that may be learned from a small amount of data using more statistical method, like certain classifier and so on, and try to
that's our
combine them.
experience too.
Yeah.
That's our experience even in some other fields other than biology as
So
well.
I think in the AI field they call this steering, right? Steering the problem. And you can also regard it as biasing the sampling towards a region that you favor and so on. So
Yeah, so definitely you've always emphasized that biology and medicine are built on the deep scientific knowledge and probably shouldn't be treated, well, they shouldn't be treated as black box machine learning problems. And in practice, right, when you talk about steering or maybe more generally, so how do you encode that domain expertise into an AI model?
yeah, so that really is dependent on the scientific question, right? So I think the general point is that biology is a very deep scientific field. There so much that has been learned by painful experiments over many decades. And so if you just throw all those away and hope that the model would somehow regenerate that knowledge, that seemed to be a little bit too optimistic, in my mind. So I think as much as possible, you would want to encode those useful knowledge some way in your model, but that is problem dependent. Yeah, it's a very difficult task. Yeah.
Yeah. And then I guess it's also related question is how do you convince maybe students or trainees who come from a pure machine learning background to do that? Or how do they do it? Right? How do you advise them to do it?
Yeah, I mean those students or postdoc who chose to work with me, I don't have to convince them to
Yes,
to
sure.
do
Yeah,
that.
yes, yeah, or advice.
They're
I
interested in
should say.
doing that. Yeah.
Yeah.
Yeah, but then they first have to build up some understanding, right, of what the true scientific question is about, and then try to come up with some idea based on their understanding. Yeah. So my role is usually just to provide some feedback on how to place the idea that the student or postdoc came up with in the larger context of because I have a longer career, so I feel that I have a broader perspective. So that kind of feedback sometimes would be helpful to them. Yeah.
That sounds great. Jing, you had a comment?
Yeah, I think it's hard to convince them if they are not intrinsically interested
I
from
take that back.
No. I think
I
it's
was
a
more
great
asking about
question.
steering. Yeah.
We are facing similar problems, right? How you steer them into academia other than like OpenAI or something like that. It's actually, I don't think there is a real solution, but I only tell them how great to be a professor. There's nothing else I can do. They may or may not be buying what I say.
Yeah. But for those who are interested,
I
right? So
often take
they will
Wing
still
as an
need guidance.
example, saying, "Wow, being a professor is so great. You can do great science. You can also have so many companies, and you can be interested in like signal processing these days. The next day you do biology and have a wet lab. That's actually great."
You can keep chasing your interest. I think that's
The freedom is
you can. Yeah, that's what's so amazing to me. Great. So next question. So in your 2023 Grace Wahba Award lecture, You discussed using deep neural networks for causal inference. So for our audience, can you explain why causal inference is a critical area where rigorous statistics can offer something that pure AI currently cannot?
Yeah, actually, I think, basically what science is doing is trying to understand
That's
causal
right.
relationship. So the term causal inference, I admire the audacity of using this term and say it's a subfield of statistics.
Statistics.
But in any case, I think nowadays this kind of causal inference that we 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 investigate in statistics, uh, they are the situation which is most useful when you don't really have the ability to do the real experiment, and then try to use some statistical model combined with you have to start with some assumed sort of causal relationship to begin with. I think if you want to say adjust for the covariate, then you sort of have to assume that those covariate that you adjust are sort of causally upstream to your predictor variable and the response variable, right? So I think this technique, is very helpful in sorting out, potential confounding and things like that. I am myself not an expert in this field. The only reason we wrote that paper is because during the pandemic, I was, hiding in my office all day and has nothing to do and decide to read up on causal inference, which is, of course, a very popular area in statistics in recent years. And so that let us, try, to sort of combine the neural network model to the question of sort of trying to so the paper that you refer to, the objective is to look at the effect of a variable X, say, on another variable Y, and try to adjust for possible confounder. And the thing is that you usually have many possible confounders, so maybe hundreds of them. And of course, you can try to adjust for all of them, but it's reasonable often to assume that there is maybe a smaller subset, right? Or maybe some feature of this large set of potential confounder that you could adjust for. And so neural network is a kind of model that would allow us to do this adjustment. So the difference between the standard AI model and this kind of causal modeling related neural network is that the latent space that drive the modeling, you want to put in some conditional independent structure, which would then allow you to fish out the small subset of feature that can affect both the predictor variable and the response variable. And then you can do your adjustment on this much smaller reduced dimension confounder. So the neural networks are there just to provide us with a very flexible function class to model this conditional independent structure and also provide the algorithm for us to do the optimization to learn those functions. So to me, as a statistician, the neural networks is just a very powerful and flexible class, highly over parameterized, but yet you can do effective, optimization and find some good local minimum that will give you, good performance. So to me, this is a tool for doing statistics.
Yeah, sounds great. Any other comments?
Well, rather than the comments, I really want to ask Vince questions about the AI and the data, because this section is for the AI revolution. Well, I think well, how we can determine whether we have enough data to answer particular science questions using AI technology. So how would that be a critical statistical or data science problem that require some theoretical framework to determine that the data science is there? Well, I may just give an example. That is not something about the big data or the AI, but that is some project I have done in Wing's lab almost 20 years, 15 years ago. By that time, Wing, Jianghui, and I tried to utilize the short-read sequencing to reconstruct the gene isoform, right? That is a very challenging and fundamental problems in the field. But finally, we find the short-read base method is very problematic. You will have a lot of false positive prediction from the short-read. That is the short-read does not contain enough information to reconstruct those gene isoforms. In fact, a few years later, Xuegong Zhang, a visiting scholar at your lab, about 25 years ago, later he proved that there's an NP-hard problem. If you only use you use short-read and relies on the MLE methods, and that would be not possible to solve at all. That's why you encourage us to use the long-read sequencing, a new technology, and the long-read sequencing contain those information. Now, revisits the problem about the data and the AI relationship. Then, well, I think everyone wants to use the AI, but not all the problem can be answered by AI if the information is not there or information is not enough. Then what's your opinion about determining those data science or information for using AI?
Yeah, that's a hard question, I think. So there are two types of exercise, right? One is that you have very large data set like the UK Biobank, and there's a lot of information there. There's a lot of science you can do with it, and you just try your best to tease out whatever useful information is there, right? So that's one way of doing statistic. The other is that you actually have a a scientific problem that you're interested in solving, and then in that situation, you have to ask yourself even though this database is very big, does it have the kind of information that is sufficient to, support the analysis to answer that question? And sometimes the answer is no. You think about it, and then you have to ask, what might be the useful data, and then try to either search for that data or to convince people to generate that data. I think, give you one example, I think we have this, large language model now applied to epigenetic data that can do a pretty good job in predicting how sequence affect epigenetic features when you are doing the prediction cross context and cross region. That means you're trying to correlate the prediction to across different regions in the genome. So that usually, give you can get pretty good performance using this AI model. But when you try to use the same, model, which in principle can predict based on sequence, can predict what would happen if you say change the DNA sequence in the promoter or in enhancer, right? So these are cross individual differences that you try to predict. Then these AI models are usually quite poor. Right? If you want to predict if the effect of personal genetic variants on gene expression, for example, it turned out that this kind of AI model is still not good enough. The performance is actually worse than if you just fit some sort of simple recursion model based on a few hundred individual, but in a region-specific manner. So I think that the reason is that if when you train this large language model to predict epigenetic features, you are usually using maybe the encode and roadmap data that are based on just a few individual or a few cell lines. So you don't actually have enough genetic variation in any region. But if you look across the genome, of course, there's a great variation in the DNA sequence, right? So I think it depends on the kind of question you want to answer, and the data may or may not be sufficient, right? The encode and roadmap data is definitely huge, right? The huge database, but for predicting, interperson, no variation is not sufficient.
Thank you, Wing.
That's great. Great question and great insight. Yes.
Yeah, I think we all learn from Wayne. It's one thing that's I think we all benefit is really root any new methods or new thinking into real applications. I also know there are a lot of people doing AI, doing large data purely by fantasy. And for example, one thing which I'm not sure is people talk about this distributed computing, and I usually ask them, do you really do that? If you do that, then real challenges would be interesting. If you just imagine there's something like that, it's hard to convince me to spend much hours and to prove theorem this is consistent or whatever. But I don't know. That's just style. I think it will be much time consuming to learn real science to understand in depth of what really is correct, what really is wrong. It's easier to imagine a situation. But for us, I think, including Kai-Fi and me and many of Win's students, we do benefit from such things, and they sort of root us. And although we can still fantasize, but we know we are fantasizing, we wouldn't mistaken that as a real application in some sense.
Cool. Yeah, so the next question actually is following up on the conversation we just had, Wing. So, but in general, generative AI, large language model that we were discussing, so some of them are genuinely transformative for the field, as you mentioned. We can they can perform very well for certain tasks, and then but also some of them are overhyped. So other than we already had a context that you gave a very good answer. So do you have anything to add as compared on what is what you think is transformative, and what you think is overhyped?
Yeah, I think the recent advances in AI is certainly very impressive. I mean, in terms of dealing with language and text data, image data, and so on. But I'm not, an expert in those areas. So my use of the AI techniques are mostly in this, bioinformatics and computational biology problems. And there I find what is useful as a sort of statistician is that this, as I said, these models provide a very rich and flexible class, although it's over parameterized, but somehow the local minimums are still very useful and very coded. And so from the advances in AI, we have these flexible function classes, and we have pretty effective optimization algorithms. I mean, those are actually traced back to stochastic approximation and so on, all statistical concepts. But somehow the computer scientists are able to sort of create very powerful programs and tools for us to use this kind of technique effectively. And finally, I think the idea of learning, embedding, and representation, using basically convert problems into vectors, tokenize using vectors. I think that's very powerful and it's very useful for a lot of problems. So I think those are really, I would say, transformative uh, ideas and highly useful in our own work also. Yeah.
Wonderful. Thank you. All right. So we're ready to any other comments? Otherwise, we move on to part 3, which is the future. So, okay. So first, you have long championed integrating population scale, genotype and phenotype data. So given the current boom in AI capabilities, how close are we to fully realizing that vision? Or what are the major statistical roadblocks that are still in our way?
Yeah, so from genotype to phenotype, it's a very long distance, right? So the DNA sequence on one hand, and then disease or development and other sort of organism level phenotype. That a very huge distance. So I have always been interested in trying to sort of partition this distance into many layers, right? From DNA to what's happening inside the cell, regulatory information and so on, then to tissue level, to organ level, organ system level, and so on. So what we have right now is that we have a lot of data on the genotype side. So, every person in principle, can have the genome sequence, right? So you have lots of personal genome sequence, and you have lots of electronic medical records or information that you collect on the person, so during routine medicine. But in between, those layers, you don't have personal level data. What you have is just reference data. So I think the key now is to spend more resources, generate more information in the intermediate layers, right? Although it cannot be truly, so population level data on this intermediate layer, but you still want to organize them in the right way. So I think technology-wise, probably we can do a lot of this. But the problem I think now we are facing is data sharing. I think this is the current situation, I think is very, suboptimal because you have to go through a lot of hurdle to get to see the sequence data and the associated intermediate or final phenotype. So that's one thing, but even worse is that we've been using, say, the GTAC data, which give you, gene expression data for many tissues and organs for many individuals. That's very useful. But last year, for example, and I say that, for certain countries, we will bar those scientists from those countries, from using this data, right? So immediately you have all the scientists in China who might be using this data effectively to do some, modeling analysis. They no longer can do that. And then what will happen is that next year, when China produce, I know that they have project very interesting that will generate very interesting gene expression data for lots of interesting primates and model organisms, and they won't be sharing it with the world. I mean, it's reciprocal, right? So this give you an example that data would tend to be, siloed. I think this is sort of the trend that is very worrisome for me. And without the ability to integrate, I mean, even if we put all this data together, generated by China, US, and other countries, it's still not sufficient. And then you have to partition it further. And so I'm not very optimistic at this point that we'll ever have the right kind of data to really bridge this gap between genotype and phenotype. The data in principle are there, and we do have enough resources, if we pull all the resources together to make some headway in generating that data. But it doesn't look like that we actually can use that data.
Well, we can keep talking about it and
Yeah.
bring the awareness.
Yeah.
Yeah. Yeah, it's a great insight. Yeah. So with the data, so assuming that there's still enough data for AI, what is your vision for the future development of AI?
For the future development.
Assuming data is not a problem.
Yeah, I'm not sure I'm qualified to say anything on that, but personally, I'm very interested in in sort of closing the loop of data-based modeling, and which is mainly occurring, so in the computer, right? Basically, it is virtual. So, but at some point, you have to make contact with the real world. So there's this term called physical AI or embodied AI. To me, that is a very important development. I think it's absolutely needed, because otherwise you cannot close the loop between experimentation and computation. Right, George Box, who is a famous statistician when I was a student, used to show us this figure. You have modeling, data analysis on one side, right? And then you have experimental design, and then you have the experiment, and then there's this loop, going through modeling and experimentation, and then you revise your model in terms of new experiment. So that ideally should be what is what's that ideally should be the approach, right? Yeah. But right now, I think the experimental side is very time consuming and not automated enough. So I think some kind of technology that would automate, at least in the genetic and molecular biology field, I see a lot of potential in trying to create systems that can automate biological experiment. Not the system should be rather general, so it can be used to automate all sorts of experiment, and not just to automate one assay. Right? There are now many automated machines to do just one assay. That's not enough, because in research we cannot predefine the experiment, right? So it has to be a rather general system. that you can use to sort of close that experiment and computation loop. Yeah, so I'm very interested in that line of research. Yeah, it's probably something I would spend some time doing in the coming years. Yeah.
Yeah, so knowing your style, I was gonna ask, is this your upcoming research,
Yeah, this
right?
would probably be my focus in the next several years at least. Yeah.
Yeah, so I think one thing I have been in China more formally for a year. I think one thing that's make you feel excited is there seems to be a lot of opportunities, like what Win said. I have been also talking with people in automations, in embodied AI type of people, and also talking to people who do protein designs, trying to establish collaborations. They
Yeah, so
need
I think the
course,
key requirement is that there has to be a system for doing automated biological experiment,
Yeah.
and that system has to be software driven so that you can, it can be directed by a program. Once that is done, then you based on your model, you can identify certain data gap that you can then design experiment and immediately write a program to get the system to perform those experiments. So and then you revise your model. So once you can close that loop, I think then hopefully there will be some some new progress. Yeah.
Yeah. And so, what do you think virtual cell, the concept of virtual cell, is considered embodied AI, or that's completely
No, virtual cell
virtual
is
experiment?
virtual, right? You're just
Yeah.
building that model for the
Yeah, yeah,
cell. Yeah.
yeah.
But I think in order to be successful in
Yeah.
building a virtual cell, I think you need this kind of closed loop learning.
Yeah.
Otherwise, I think it's pretty hopeless.
Yeah. Wonderful. All right. That was a great discussion. So moving on to part 4, about mentorship and advice for the next generation. When you already gave a lot of advice and really inspirational conversation already, but yeah, so here are a few more questions. So you have an incredible legacy with over 270 academic descendants. And then you've often said that, as you mentioned, interest trumps everything else. So how does that philosophy shape your approach to mentoring, and how do you guide your young researchers who are trying to find their genuine interest between traditional statistics and the rapidly moving AI space, and perhaps the computational biology?
Right. So since you bring up my large number of students and collaborators, I just want to take this opportunity to say that I was watching Conan O'Brien's Harvard commencement speech, which occurred 1 week ago.
which one?
And so I just want to
Ron-
quote
Ronnie
one
Chan?
of the lessons that he learned
Oh.
is to always remind myself that I have done nothing, absolutely nothing alone. So I think, this is my feeling. I mean, all my work, I just look at my CV. I think I have about 250 papers, and only 8 of them are single author by me. So almost all my work. I mean, I think so. What Conan said is absolutely true. So I just want to say how lucky I am to have all these wonderfully talented students and wonderful collaborator that together, um, was able to do some fun science. So just want to say that. So regarding the way I mentor students. I think I said it before. I think my view is that the student has to have interest
Okay.
in the problem that they work on. So all I can do is to say, well, this sort of general direction may be useful and interesting, and they would really have to go in and find out which aspect is of particular interest to them.
Mhm.
And now, once they have done that, then if they come back to me, I hope I would be able to provide, some perspective because of my broader context. I know the scientific landscape better, so I would try to help them to put their specific interest that they identify in the context of this broader scientific landscape,
Mhm.
and then iterate, right? Then they will go back and try to do more thinking. So so that's sort of my my overall philosophy in terms of mentoring. And the other thing is that I try very hard to connect them to people who are doing interesting science. Um, and in a place like Stanford and Harvard or Chicago, there are always many interesting scientific activity going on, and it would be a pity not to take advantage of them.
Mhm.
So that's the other kind of mentoring uh, activity I
Mhm.
I pursue. Yeah.
Yeah. I think that this I have to ask my discussants here to
Yeah.
comment on.
Well,
Yeah,
I
anything to
well,
add?
Wing, I think it should be us who should say thank you. Yeah. Yeah, actually, what Wing said just now exactly happened to me. Exactly happened to me. Well, saying that, Wing helped me to really shape how we can utilize the long way in the transcriptomics analysis, right? I think back to 2011, I think it's in early spring, something like that. We have such an idea, and then, well, of course, by that time long, we got a lot of errors, something like that. The throughput is low, and then I met Wing at least about maybe 5 or 6 times in the second half of 2011, and Wing helped me to figure out, okay, what would be the key questions, okay, the robot to work using the long read sequencing. I do remember the very last meeting right before the Christmas. I went to Wing's office. I stayed 2 hours there. That would be the very last meeting before I went to Orlando for my Christmas holiday. But at that time, I think we figure out, okay, that would be the very, very last technical problem to about using the long sequencing. Okay, then, okay, we are ready. Then Wing helped me to establish the collaboration with the pepper company. Okay, that's how we can generate the data and also send an email to the director in the Stem Cell Biology Institute at Stanford, uh, who can generate those cell for us for to generate data. So I think that's really the whole process to build up a mindset for independent research, not only for a project. Okay, I think in this process, I learned, okay, how I can ask a meaningful question and how I can solve what Wing says partition, right? Partition this role into a small pieces question, and then I solve it one by one, and then I finally can achieve that goal. Yeah, I think this is really an important process for young scientists to work an independent scientist. I trained me to become a professor. So when we talk about his principle of training a student, a postdoc, we might very many meetings that we met in that years. I really make it possible and achieve my dream to become a faculty. Thank you so much, Wing.
Yeah, my feeling is that I learned a lot from Win like we discussed a lot of problems. So at least when I was close to Win, when I was students, we always sort of whenever a new problem come up, and Win will pose a problem. At that time, there's less use of computer, usually a piece of paper. Then we start to discuss. I think I was amazed quite a few times by the way like Wayne approaches a problem. I think he never really said to me saying, "Oh, you should do this, do that." He will just write it down, and I would just watch it, and then said, "Oh yeah, let me do this. I can go to do something like that or something like that," and 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 he, I think I sort of watch him to learn biology and to start to approach biology problem. I feel inspired. I will try to start discussion. And when was tell me so which maybe interesting areas, which may be exciting, like microarrays. That's the one time I remember he explicitly told me saying you probably should go to meet Pat Brown or something like that. And because I was at Stanford, I was visiting Wayne at UCLA. So it's not really directing anyway, right? More like friends chatting and saying you should probably look up certain things. It's very interesting and in a way, I think that's probably Wayne style. In Chinese, it's called 润物细无声哈. It sort of influence us in the way
How do you
he's
say it in English?
hard to translate into English, right?
Oh, yeah, a little drop of rain.
Yeah, it's a
Yeah, great.
yeah. We did because the way he is always at the sort of very new and different areas. I think without saying, we sort of feel quite one way it's inspired, also another way it's very embarrassed because your teacher is already doing new things. You're still doing old stuff, something like that.
So to summarize, let me try to summarize. So with the rapidly moving AI, right? So how the mentorship style, like the key mentorship style, would still work. As you know, we learn from Wayne is to follow to if we see an interest and a spark from the trainee, we focus on uh, making the spark, turn the spark into a fire and contributing the necessary materials along the way, right? To see to make it grow. Is that a proper summary in a very abstract way?
That's pretty good, yeah. So yeah, I would just like to add that there's this very famous saying by John Tukey. He said the best thing of being a statistician is to be able to play in everyone's backyard.
Backyard. Yes.
And so I would supplement that by saying that, and if you are lucky, sometimes you would get to grow a tree in someone's backyard. And that's sort of what we, should aim to do, right, when we get into a scientific area. Yeah. Not just playing.
Yeah, yes, yes. Love it. I love it. Growing a tree in the backyard.
The little
Excellent.
drops grow trees.
Little drop of rain, grow
Ah.
trees. Yeah. Okay, all right, that's great. So next question. So in an era where data is abundant, well, I know it's not abundant enough, but let's say it's for some area it's still abundant. But the true domain expertise takes years to develop. So how should early career researchers balance learning the newest machine learning methods with learning the underlying science deeply enough to sustain a decade-long career?
Well, I think everyone is different in the way they allocate their time and effort. But I would say in general, you can probably afford to keep both of these things in mind, and maybe you can concentrate on one of this or the other when you see something really interesting and exciting, right? If it very interesting, exciting things happening, and you're interested in this say new type of machine learning, then you may spend majority of your time catching up and thinking about it, right? And likewise, if there's some new development in biology, like CRISPR suddenly appear, and and
That's right.
you have collaborator that is generating interesting data using that, then maybe you should spend more of your time doing that. So I think but it's good to always keep both in mind. In the back of your mind, that you need both.
And then it will be a project or question specific.
Yeah, right.
Got it. I'll ask the other two professors. I mean, you have your trainees. What's your advice? Anything to add?
Well, Jun,
Yeah,
please.
go ahead. Okay, you can go ahead.
No, I don't know. Well, but I think I always ask my trainee to learn how to ask a meaningful question, as I said just now. Right? We got tons of paper every day, but you want to make some noise in the field, right? So you don't want to spend many days in some things that is not meaningful at all or impactful at all. You want to, well, those questions could be low hanging fruit. I'm not saying that the meaningful questions are always very hard ones, but so that is something I really learned from Wing, and this is almost the very first lesson when some trainee come to me and say, okay, Kim-Fai, I want to stay in academia. I want to become a faculty in the future. And I say, okay, well, in the upcoming few years, well, you need to find meaningful questions. Of course, I would be very happy to work with you at the very beginning, since the first project, right? To find a good topic to work with for 1 year or two. And then you need to figure out some meaningful question by your own. This is exactly what Wing did to me. Well, I published the first paper in Wing's lab for this RNA-seq, the spice map, the RNA-seq aligner. That is an idea given by Wing. And then it's the second one, and then Wing asked me to think about those meaningful questions, and then I didn't do anything for about half years or even longer than that. It's not productive at all. But I just keep reading paper, and then discuss those ideas with Wings, and then finally find a good question to answer in the second half of my trainees. But so that is something that I pass the lessons, and it's something I learned from Wing to the next generation. I want to let you know that I just a few hours before this conversation, I received an email from Boston, from first trainee, first postdoc in my lab, the first member in my lab. He said, thank you, and say hi again and keep me updated. And he established a new staff companies in Boston. I think that is how the trainee really appreciate mentorship. Yeah. Again. Oh, by the way, my first postdoc joins Shelley Liu's lab, who was the former students in June's groups 25 years ago, right? Yeah. Oh, wow, a small group loop. Yeah.
Yeah. I think when you have a very generic question, anytime
it was.
in your life, you probably face same question. It's not maybe AI
No.
versus science. It's maybe something else, right? But there are always a trade-off
The
one way or another.
balance. Yeah.
But for AI, I think as Wayne said earlier, for any of this, it's driven by interest. If you are so strongly interested in biology or in whatever science, you would definitely be happy to learn more. There's no sacrifice or whatever. You actually feel happy to learn them. But if you feel you are sacrificing something, you probably should find something else to do.
Oh, yeah.
You should do neither, maybe.
That's true.
Yeah, but otherwise, if you feel strongly, say, to do theory and for AI, I think you need to learn. It's a tool, right? It's almost like a language these days. If you're able to use it more effectively, you get more efficiency, like in getting the
That's right. Then you should have more time to learn.
reference, getting those things. And the other day, I talked to Tony Tsai. He said even it's great to actually. It's not necessary to ask AI to review paper for you, but you can ask AI to review your own paper and
That's right.
very interesting referee comments. You actually feel it's helpful. You can revise your paper accordingly. I think it's interesting thoughts. And yeah, but for us, that's not really AI as a technical thing. It's AI as a daily use tools, which may help you, right? It's like using typewriter, things like that. But for me, I'm very curious about how AI works, so I do want to spend more time to understand the
Me too.
details.
Yeah.
Like one thing which amazed me, which I regret I didn't pay attention earlier, was that they have this everything nowadays AI training is about this score matching ideas, like doing the score. If you don't know it, don't worry about that. But actually, this is related to one of the very early problems we encountered when I was student with Wing. As doing statistical inference with this unknown normalizing constant, where normalizing constant may involve some parameters. We are so stuck with the likelihood methods. It's actually it made me rethink statisticians as a whole. So we have been trying that problem for many years. Like another thing is the density estimation. We have been doing density estimation since ever, but what progress have we made? Not too much. I mean, theory, sure, but practical impact is quite limited. But all these problems are very statistical, and this score matching ideas, which actually works great, and it's becoming a foundational tool for AI. So from that perspective, I actually like to see how AI works, and then you realize more and more it's not really we are bragging or anything. It's really statistics. It's not a lot of these fundamental methods are very statistical. Even it's not invented by statisticians. Techniques may not be invented by statisticians, but it actually give us some lesson to learn, saying, why didn't we think about that earlier? Right? All this generative model is really about density estimation, right? So where have you stuck with 2 dimensions? They're doing like 2 million dimensions or whatever. But also it poses questions which we still cannot solve. What is really generative model doing? So by sort of imputing, generating new images or whatever. So it must be doing some kind of imputation or interpolation, but we are not able to describe it mathematically what it is doing. But it's interesting to me.
Great comments. So we're moving towards the end of the conversation. I'll save one last question for Wayne, but before that, just a fun question. Don't take it too seriously, because I know it's a tough one. But I'll ask all 3 of you. I'll start with Jing. If you were starting your PhD today, what problem would you work on? And Wayne, you will be last.
What would I be working on? I probably will work
For
on
fun,
AI.
just for fun.
I think I will probably for AI-related problems.
No biology or AI in biology
No, I
or
started my PhD. I don't know anything about biology. Maybe I could do some biology as well these days, but it's hard to imagine. If I go back to my years, just move the time. I was as naive as I was being a PhD student with Wing. I probably still work on AI-related theory because that's what I did. I choose this give something related theory, sequential Monte Carlo methods. I didn't touch anything about biology, although both Wayne and Augie were working on genetics at that time.
Yeah, all right. King Fai?
Well, yeah, it's AI related as well. I would say is that biology is a type of science very different from the physics or the other chemistry. maybe.
So what was your training, Kai? I forgot to ask.
Structural.
But I got a degree of biology in Tsinghua University.
Tsinghua biology,
Wait,
I see.
no,
But
PhD
I
stuff.
got a PhD in biophysics, structural
Oxford.
biology. So I also study protein structure, and that is motivate myself to develop software because by that time we use a lot of software to predict and analyze the protein structure. And I have a stronger interest of developing those software than utilizing those software. But I think how to utilize the AI to organize the data or to analyze the data is maybe an appropriate way to study biology. Would be
But yeah, so sorry, the question is, what would you work on if you do your PhD now?
well, okay. Saying that what my PhD project will focus on how to compress the large amounts of the NCBI data. Okay,
Oh,
okay.
okay.
We train a bigger, well, the data is there, right? The
That's AI.
information is there. Well, how can we discover those information that nobody utilized in the past? Well, we know many paper published a huge numbers of data there, but they only analyze a small part of them, right? Well, how about the others?
Large
Information.
data.
The information is there, but nobody utilize those. Well, we don't want to put it in the trash bin, right? We want to recycle them, I would say. Well, then AI would be a good technique that we can utilize, and yeah, that would be my
Your
I
PhD.
yeah,
All right,
PhD
okay.
project.
Okay, win.
Yeah,
It's a fun question. It's not too
I'll
serious.
probably still be working in the area of the statistic plus something, right? So what is that something? I think probably the area of sort of automation and synthetic biology. So it's like closing this loop between computation and design the experiment. Well, it's natural because that's what
Yeah.
I'm currently interested in. So
Maybe can you describe a synthetic biology a little bit for the audience?
yeah, basically you would basically design, let's say, genetic circuitry to maybe sense something, right? You have basically a molecule that would changes conformation or induce some kind of signal depending on the cellular environment, right? And then another part of the circuitry would then respond to that once you detect that signal and so on. So basically you have artificially designed subsystem that you kind of instill into the cell. And there's basically once you can do that, then it open up a lot of space for new discovery, I think.
Sounds great.
Mhm.
Yeah, I guess, yeah, if I were to do my PhD again, I would do still statistics plus biology. Yeah, which
That's very
is
general.
just like yeah, 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 maybe plus AI. But yeah, great. So one last question for Win. Um, so for the also the audience, the next generation, what is the one thing that you wish you had known at the beginning of your career?
Oh, well.
Or many things, I don't know, or nothing.
I think maybe you know maybe it's a lot not to get depressed or frustrated when you get rejected.
Mhm.
Right? Rejected for a paper, rejected
Yeah.
for a proposal, but just try again, move on, and don't get hung up on that. Yeah, I think it save a lot of agony.
Sounds good. I'll remember that. Great advice. Yeah, so and then with that, do the discussants, do we have any other comments, thoughts? All right. Great. Thank you.
Thank you, Wenyi.
Thank you.
Yeah,
It's a
yeah,
very interesting session.
yeah.
Thank you. Thank you very much. Thank you for having me.
Yeah, thank you.
Great
We'll see
fun.
you in the JSM then.